Welcome back to Biology Boost Up. Today in this new video lecture, we're going to learn about the enzymology or the enzyme. So now let's start. Now look the definitions of enzyme. Basically the enzyme is a biocatalyst synthesized by living cells. They are protein in nature, colloidal and thermolabile in character and specific in their action. The enzyme producing glands are called exocrine gland, and the enzyme transported by the ducts, and it can be stored inside of the gland cell. Now focus on the chemical nature of enzyme. Okay, all the enzymes are invariable proteins and it has its own tertiary structure and specific conformations. The functional unit of enzyme is called the holoenzyme, which is often made up of apoenzyme and cofactor. Basically the apoenzyme means the proteinaceous part of the enzyme and the cofactor is the non-protein part of a enzyme. On the basis of chemical compositions, the enzymes are classified into simple enzyme and conjugated enzyme. Now the simple enzyme is made up of only proteins, whereas the conjugated enzymes made up of protein and non-protein parts. Normally three kinds of cofactor may be identified. First, the prosthetic group, second is coenzyme, and third one is metals ions. Now the prosthetic group. The prosthetic groups are the organic compound and they are tightly bound with the apoenzyme. Now look at the coenzyme. Actually those are also organic compound, but their associations with apoenzyme is only transient. And the metal ions, they are for coordination bond with the side chains at the active site of enzyme. Now let's talk about the properties of enzyme. First, the enzymes are secreted from exocrine gland, acts on specific substances and remain unchanged after its reactions. In the second point, the enzymes are active in small quantities but accelerate the chemical reaction. Hence, the enzyme is a biocatalyst. In the third point, the enzymes are highly specific. That is, an enzyme will generally catalyze only one particular reaction. In the fourth point, there are close relationship between the shape of a active site of enzyme and the substance's shape. In the fifth point, all enzymes are thermolabile and colloidal and proteins in nature. As you can see here, the enzyme has a place where the substance can bound. That place is called the active site. And when the substance comes and combined with the active site of a enzyme, then this form a enzyme substrate complex, and after this complex formations, the enzyme catalyze those substances and produce the product of that substances. Now let's talk about the functional role of enzyme. Some enzyme based technique used for detections of disease like AIDS, malaria, etc. Second point, enzymes are used in breaking DNA at specific site. That fragments are employed in genetic engineering. In the third point, enzyme complex zymogen, which is obtained from yeast, is used in formations of alcoholic drinks. Now in the next point, in humans or other animals' body, some enzyme helps to digestions of different foods, like pepsin helps to digestions of protein. Next point, some enzyme act as a antiseptic to prevent growth of disease causing microorganism, like lysozyme present in saliva and tears. Some enzyme control the cell cycle, DNA replication, transcriptions, as well as protein synthesis and different biochemical activities which is held inside the body. And the next point, the streptokinase is an enzyme which is used in cleaning blood clot inside the blood vessels. Thanks for watching.